Welcome back, students. Today we are going to discuss neural control and coordination topic. This particular topic belongs to Unit 6, that is multicellularity, structure and function, animal life. Today we are going to discuss part 1 of this particular topic. Before we start the main content, I would like to inform that the images used in our slides are for education purpose only. We respect intellectual property right of the original creator and owner of these images. If you are the owner of any of the images used in this PPT, have any concern, any issue, please contact us at sathee@iitk.ac.in and we shall remove the images. In this particular video lecture, we are going to discuss these points in both the part 1 and part 2, a nervous coordination, central nervous system, automatic nervous system, peripheral nervous system, receptors, effectors, reflex action, basic physiology of special sense organs, integrative control by neuroendocrine system. Okay, so these are the topics which we are going to discuss. First of all, in our body, means in human body, the coordination of organs and its organ system is vital. Why? Just to maintaining the homeostasis in our body, right? Ensuring stability in internal conditions, right? So this coordination actually involve interaction and complementation of functions among various organs. Okay, so let's understand how this is achieved in our body, how nervous system help us to achieve the important type of interaction which is actually required by each cell of our body. Okay, as you know, in our body we have so many organs and organ systems, right? And this system actually responsible for control and coordination is nervous system. That kind of system which is mainly involved in the control and coordination mechanism of our body. That system is known as nervous system of our body, right? So if we broadly divide this nervous system, we can say that nervous system are of two types. First one is CNS, okay, or we can say central nervous system. Another one is PNS or peripheral nervous system. This CNS or central nervous system include brain and spinal cord. Remaining other type of nerves or nervous system comes under peripheral nervous system. Okay, so nervous system we have divided into two different categories, CNS and PNS. Clear? So this CNS, as I just now told you, this CNS is composed of brain and spinal cord, while this PNS is composed of various other veins. Okay, so we can further divide nerves into two different categories. First one is afferent nerves or afferent fiber, which mainly involve to transmit impulse from the various organs towards central nervous system. As you can see over here, this afferent nerve involved to transmit the impulse from skin to the central nervous system side. Second type of nerves are known as efferent fibers or efferent nerve, which actually involve to transmit a regular impulse from central nervous system to the affected area or affected organs. Like in this case, this is central nervous system and this is any organ, means in this case muscle. If transmission occur from the central nervous system towards the affected area means towards muscle in this case, that time we can use the term efferent nerves for the same. Okay, so two type of nerves are there, afferent and efferent. Work just opposite. Afferent involved to transmit impulse from the organ towards the central nervous system side, right? While efferent just perform its work, vice versa. Clear? Now, peripheral nervous system, we can categorize peripheral nervous system into two different categories. First category is somatic nervous system or neural system, and second ANS or autonomic neural system. Okay, central nervous system have two parts. Just remember, brain and spinal cord. Second one, peripheral nervous system. Now peripheral nervous system further categorized into two different categories. First is somatic, another one is autonomic. Okay, somatic actually rely impulse from central nervous system to the skeleton muscles. Where? Skeleton muscles. Okay, while this autonomic neural system transmit impulse from the central nervous system to the involuntary organs and smooth muscles. Involuntary organs means heart, lung, stomach. They all are involuntary organs who don't require our wish. Okay, all those organs which continuously perform their function. Okay, so this autonomic nervous system actually transmit their impulse from central nervous system towards involuntary organs and smooth muscles. While here, this somatic neural system rely impulse from central nervous system to the skeleton muscles because they are in our control, right? So let's discuss their function. As this is the skeleton muscle, this is again a skeleton muscle. Okay, this is brain and this is the spinal cord. These two are the part of first part, right? Now they transmit their impulse from brain upper motor neuron to the somatic neuron side, and then through lower motor neuron towards the skeleton muscle side. And similarly, this via directly or via spinal cord. Somatic motor neuron or spinal cord through skeleton muscle. Okay, so this is how they actually transmit their impulse from central nervous system to skeleton muscle side. Now, automatic neural system. This automatic neural system further divided into the two different categories. First one is sympathetic, second one is parasympathetic. Okay, sympathetic and parasympathetic, they also have opposite work. Like sympathetic nervous system, if involved to increase some kind of function, like increase heart rate, increase hormonal secretions of stimulate some kind of glands, right? So parasympathetic just do opposite work, decrease the heart rate, inhibit the secretion of certain hormones. So one by one we will discuss it. Like the sympathetic nervous system involved to dilate pupil of our eye. It inhibit, mostly it accelerate the function or stimulate the function. One or two function, its function is just to inhibit the function. Like in the case of salivation, it actually inhibit the salivation. But normally, like it involved to dilate the pupil, increase the heart rate, dilate our bronchi. Okay, stimulate the breakdown of glycogen. It sympathetic nervous system involved to stimulate the secretion of adrenal and noradrenal gland. While some functions like salivation is inhibited by the sympathetic nervous system, digestion is inhibited by the sympathetic nervous system. Similarly, bladder contraction is inhibited by the sympathetic nervous system. Okay, but mostly it mainly involved to increase or stimulate the functionality of various kind of organs. While parasympathetic, which is the second category, second type of the autonomic nervous system, it involve just opposite work of sympathetic nervous system. Like sympathetic involve to inhibit, so here it actually dilates, so here it involve to contract. Okay, it inhibits salivation, it parasympathetic stimulate the salivation. In that case, parasympathetic involved to increase the heart rate. Here, it slow down the heart rate. Okay, similarly, contract the bronchi, while it stimulate the digestion, it stimulate the bile secretion. Cause of to bladder contraction. Okay, so this is how we can compare both the, if you learn one type of neural system functionality, you can just write down just opposite that functionality is performed by the another system, like in this case parasympathetic. Okay, now our nervous system have so many parts. This is the brain. Okay, this is the brain, then spinal cord. Okay, and then several nerves which are involved. Okay, so this is the picture of our brain. It is having so many parts like frontal lobes, parietal lobe, occupational lobe, cerebellum, medulla region is present over here, pons region is present over here. This is the temporal lobe, hypophysis present over here, hypothalamus region is present over here. This is thalamus region. Okay, these are certain parts of our brain. Okay, corpus callosum, central calcus. So these are the part of our nervous system. Okay, so one by one we will discuss all of this. And first of all, we have discussed this functional unit of neural system. That is, what is the functional unit? Neuron. What is the functional unit in this case? Neuron. So let's discuss the structure of neuron. This is a general structure of a neuron. This is the first part, which is known as cell body, bigger part. With this cell body, so many branches are associated. These branches are known as dendrites. There is a long branch which is known as axon, is also linked with this. And the end of this axons, there are certain synaptic knob-like structure, synaptic, which is known as synaptic terminal. And in the synaptic terminal, certain type of neurotransmitters are there present in a vesicle. Okay, so this as you can see in this particular structure, in this particular cell body, it is a kind of cell. Okay, so it is having a very well developed nucleus, very well developed endoplasmic reticulum, right? Golgi apparatus, mitochondria. So all the important organelles are present over here. Okay, and this cell body is also having cytoplasm, and this cytoplasm is having, as you can see over here, there are certain bluish grayish dots are present over here, and these dots are known as Nagell's granule. Nagell's granule. Okay, so cell body contain cytoplasm with all the organelles and Nagell granule, and with this dendrites are associated, as just now we have discussed, and these dendrites are short fibers branching from cell body, and it actually transmit impulse towards the cell body, means from dendrites to the cell body side. Okay, so it involved to transmit the impulse towards the cell body. Third part is axon. Third part is this axon. Okay, so first part cell body, second part dendrite, third part axon. It is a long fiber with branched distal ends. Clear? Which actually transmitted as a synaptic knob. These are the synaptic knob, which contains, as I just now told you, which contain neurotransmitters. And these neurotransmitters are present in the certain kind of vesicles. Okay, so this is the structure of a typical neuron. On the basis of the presence of their branches, we can divide neuron into various categories, like multipolar neuron, bipolar, or unipolar. Okay, so multipolar means, as you can see in this particular picture, one axon, this is the axon, one axon and two or more dendrites. As you can see, two or more dendrites. Okay, this type of neuron is known as multipolar neuron. And this type of neuron is mainly found in the cerebral cortex region. Second type is bipolar, in which one axon and one dendrite. One axon and one dendrite. Clear? And this type of neuron is found in retina. Retina, our eyes retina. Third type is unipolar. Unipolar means this is having a cell body with only one axon. Cell body with one axon. And these are mainly found in the embryonic stage of. These are found in this type of neuron, unipolar neuron, where cell body with one axon is present only. Okay, this is known as unipolar neuron, and these are found in the embryonic stage. Now, type of axons. Type of axons, two type of axons are present, or we can classify axon into two different categories. First one is myelinated, in which myelin sheath is present, and another one is non-myelinated, where there is no myelin sheath is present. Myelinated is we can say that in this type of axon, Schwann cell is having Schwann cell form a sheath which is known as myelin sheath. Okay, so if roughly I just draw a diagram for you. This is a cell body, say for example, and this is a axon having synaptic knobs various. So if like this, it's a rough diagram. If this is the myelin sheath, okay, so this is the myelin sheath. This is known as Schwann cell. So if Schwann cells are present and Schwann cell is secreted a sheath which is known as myelin sheath. If myelin sheath is present, that time we can say this axon are myelinated. And this gap is known as this. There is a certain kind of gap, which this gap is known as nodes of Ranvier. These are the gaps which is present between the myelin sheath. Okay, so this type of axon is known as myelinated axon, and this type of axon are found in spine and cranial nerve, where fast conduction of impulse is required, fast conduction via myelin sheath. But in some other type of axon, Schwann cell are not able to synthesize this myelin sheath. So Schwann cell doesn't have myelin sheath. Those type of axon is known as non-myelinated. So this type of non-myelinated axons are found in autonomic or somatic neural system. So this is the difference between two, where Myelinated axon are present where fast conduction, fast impulse is required, like a spinal cord and cranial, while autonomic or somatic neural non-myelinated axons are there because Schwann cell are present without myelin sheath in this case. Okay. So now generation and conduction of nerve impulse. So generation of conduction of nerve impulse in a nerve, normally nerve was present in a situation which is known as polarization. Neural polarization, in which neurons are having certain kind of polarity, right? So neurons have polarized membrane. Its membrane is polarized one, which is actually crucial for the excitability, because conduction or impulse occur through nerves only. Okay. And when current is passed, when there is a polarity, means opposite charge are there, and if um, opposite type of charge, if some one place more negative charge, another place more positive charge, right? So the in this case, in nerve, the membrane of a nerve maintain a resting potential in normal case, due to selective permeability to certain kind of ions like potassium ion. Okay, so the membrane, if this is the membrane of a nerve, it is having certain kind of ionic concentration inside the nerve and other type outside the nerve. As inside the nerve, potassium ion concentration is more, while outside the nerve, normally concentration of sodium ion is more. Inside the nerve, concentration of some other negatively charged ions like phosphorus, sulfur, and negatively charged some proteins, negatively charged are present more inside the nerve. So that's why if we compare this with the outside, means extracellular fluid side, extracellular fluid, and this is the exoplasm. Exoplasm means cytoplasm which is present inside the nerve, inside the axon. What is this? This is axon, right? So this is the exoplasm. Okay, so concentration of potassium is more inside the exoplasm if we compare it with the outside extracellular fluid. So, and there are certain kind of channels are present on the axon, like sodium potassium pump, like some voltage, some pump which is mainly responsible for protein, right? So there is a selectively permeability for potassium ion and nearly impermeability for sodium ion. Means potassium ion are able to go outside, but there is, in normal condition, in polarized state, there is not possible sodium ion to come inside. Okay, get it? Now, so this polarized state, polarization, is upheld by sodium potassium pump, which actually maintain ionic gradient, on both the side. Clear? So now let's discuss the mechanism of nerve impulse. You just with a full concentration, just see this animation. This red zone showing action potential. This yellow one is the normal polarized state. So in normally, what is the charge present inside the axon? This is negatively charged, right? While outside, positive charge. Means inside more potassium, phosphorus, sulfur, and some negatively charged protein. right? While outside, more concentration of sodium ion, more concentration of and less concentration of potassium ion. Here, more concentration of potassium ion. Obviously, when it is nearly impermeable for sodium, so very low concentration of sodium is present inside. But during impulse generation, some gates are open, which allow to come sodium inside. Sodium come inside. So what will happen? Sodium ion come inside. So if we compare it with the outside, now concentration of sodium ion or positivity will increase inside the axon as compared to outside. So charge has been changed from negative to positive, and this time we can use the term action potential. Pulse will generated. Clear? So when a stimuli or stimulus is applied, this membrane, exoplasm membrane, this membrane become permeable to the sodium ion. Okay, leading to depolarization. Normally, this yellow structure is known as polarization. And this red one is known as depolarization. So when there is a membrane permeability occur for sodium, as sodium ion is present more in the extracellular fluid, so sodium ion very quickly come inside, and this will convert this polarized state into the depolarization, means generation of action potential. Generation of action potential. Clear? So this depolarization propagate along the axon, as you can see over here, from this to this side, from this side to this side. So sequentially allowing for a conduction of nerve impulse. Allowing for the conduction of nerve impulse. Clear? So as you can see in this particular picture, very clearly, this is a, what is this? This is axon. What is this? This is axon. These are Schwann cells. This yellow one are Schwann cells. Clear? And there is a notch. This notch is known as rod knots of Ranvier. And this is a myelin sheath. There is a myelin sheath is present over here. And generation of voltage or impulse occur in this direction. Normally in polarized state, what is the situation? This is the situation. Potassium ion more inside the sodium ion less, more phosphate, more sulfate, more negatively charged protein, right? So as the conduction start at similar time, depolarization, this particular some gates are open for the sodium. So very quickly, but for a very short period. But in that short period, sodium very quickly come inside. And it will change the, when it come inside, then obviously concentration of positively charged ion and if the concentration is high over here, then obviously through diffusion, high concentration to lower concentration, diffusion occurs. So very quickly sodium ion come inside, and very quickly what happen? Charge will change from negative to positive, and this situation is known as depolarization. And this is how generation of voltage occur. So impulse will generate and move from polarized to depolarized, then repolarization, then depolarization. This is how conduction of impulse occur. Okay, so depolarization is swiftly followed by repolarization. Now what is this repolarization? What is the normal stage? Polarization. First polarization, which is a normal situation which is present in a nerve. First stage, polarization, which is a normal situation present in a nerve. Am I right? Okay. So first and then depolarization is the second step, where some gates are open and very quickly sodium come inside. Okay. And then from depolarization, repolarization occur. Here we can say repolarization. Repolarization. In which again that gates are open, right? And very immediately this potassium ion diffuse out. As you know, this potassium ion is present in more concentration inside the, again gates are open for potassium, so again diffusion occur from the high concentration to low concentration. So very quickly this potassium ion just move out from exoplasm to extracellular fluid, and this is how it will again restore the resting potential. So first stage polarization, followed by depolarization, and third repolarization. This is how conduction of nerve impulse occur. Okay, so this ensures the neuron is responsive to further stimulus. The sequence repeat along the length of the axon, allowing for swift transmission of nerve impulse. Swift transmission of nerve impulse. What is the normal resting potential? That is minus 70 millivolt. What is the action potential? That is plus 30 millivolt. Okay. Now, when the nerve impulse conduct from the axon very fast towards the end of the axon, and this is the, as you can see, this is the axon, this is the end of the axon. This end is known as synaptic knob. These are the synaptic knobs, and in this synaptic knob certain kind of vesicles are there which contain certain neurotransmitter. These are synaptic vesicles. And what is this outer covering? This outer covering is known as presynaptic membrane, and this is the another membrane, this of another axon. This is known as postsynaptic membrane. These are very much close to each other, right? So there is a synapses form. What is this synapses? Synapses are the junction. Synapses are the junction between neurons. Okay, between two or more than neurons, where nerve impulse are transmitted. Okay, so this is presynaptic membrane of one neuron, and this is the postsynaptic membrane of another neuron, but these are too close. So very quickly these vesicles, synaptic vesicles, come towards the membrane site of presynaptic membrane, fuse with the membrane, just release the neurotransmitter, and these neurotransmitter very quickly attach with the receptors which are present on the postsynaptic membrane, as you can see over here. Okay, and this is how conduction of impulse occur. Okay, so if we classify this synapses are of two types. First is electrical and another one is chemical. Okay, normally when these neuron membrane are in close proximity, as you can see in this picture, these are too much close proximity. Two neurons are in close proximity. This allow direct flow of electric current from one neuron to another. And this is how impulse transmit from one neuron to another. And this is similar to conduction of just like impulse conduct from one neuron to another, like impulse conduct within a single axon. Okay, these are so close to each other. Second type, so this is known as electric synapses. Another one is chemical synapses. In this chemical synapses, neuron membranes are comparatively far apart. These are not in close proximity. So there is a gap. That gap is known as synaptic cleft. In this synaptic cleft, there is a fluid is present. Okay, and there is a fluid is present. So this is different from there is not a direct conduction, just like axon, as just now we have discussed for electrical synapses. Electrical synapses is comparatively fast because there is no gap, no synaptic cleft. But this is rare in our system too. And normally, when two axon synaptic knobs are comparatively, there is a gap, synaptic cleft like this, then chemical synapses, through via chemical synapses, impulse is transmitted from the one neuron to another. Initially, action potential that is generated in the axon, very quickly it reaches towards the presynaptic terminal. This is the terminal region, synaptic knob. This is the outer covering. Okay, presynaptic terminal, covering of one neuron. This is the postsynaptic channels, and we can say this is the postsynaptic, this one, postsynaptic membrane. Okay, so very quickly this action potential reach towards the synaptic terminal. Okay, and then depolarization of presynaptic terminal. As depolarization occur, it will open the calcium ion channels. Calcium ion channels are also present over here. So very quickly calcium come inside the synaptic knob, and this calcium ion is actually required to trigger the release of neurotransmitters from the vesicles. As you know, these neurotransmitters are present in the vesicles. This calcium ion is actually trigger the release of neurotransmitters from the vesicle. Very quickly these vesicles come and attach with this, fuse with this membrane and release these neurotransmitter into this synaptic cleft. And via synaptic cleft, these neurotransmitter very quickly go towards the receptors which are present on the postsynaptic channels. Neurotransmitters bind to the receptor site present on the postsynaptic membrane. Then fifth point, opening and closing of channel cause change in postsynaptic membrane potential. Obviously, when there is a diffusion occur from high concentration to low concentration, then change in postsynaptic membrane potential has been changed, and then action potential propagate through the next cell. So this is how action potential or impulse conduct from one neuron to another neuron. Okay, and in the last, these neurotransmitter which actually bind with the receptor is inactivated or transported back to the presynaptic terminal when the impulse pass from one axon to another neuron site. So this is how conduction of impulse occur via chemical synapse. Clear? So impulse transmission process, axons, these are already points which we have discussed, but just now very quickly I'll read it out. Axon terminal contain vesicles which is filled with the neurotransmitter, as you can see just now in the picture. When an impulse, which is known as action potential, reach the axon terminal, vesicle move towards the membrane. Yes, then vesicle fuse with the membrane, correct, and immediately it release neurotransmitter into the synaptic cleft, and then neurotransmitter bind to the specific receptor which are present on the postsynaptic membrane. Ion channel open, allowing ion to enter into the postsynaptic neuron. Now new potential, it may be excitatory or inhibitory. Inhibitory, GABA comes under inhibitory. Acetylcholine comes under excitatory, develop into the postsynaptic neuron. So this is how impulse conduct from one neuron to another. So this is all about the part 1 of nervous system. I hope most of the things has been cleared now. If still you have any doubt, any confusion related to today's video lecture, please let me know. We will discuss it later on. Till then, take care, bye-bye, and keep learning. Namaskar.